Welcome back to our series of videos on the nervous system. In this one, I'd like to explain how an impulse travels along a neuron, or some people say it's within a neuron. The topic of how it goes from one neuron to another neuron is a totally different topic, so I'd really just like to focus on how an impulse travels along a neuron. And I've presented this picture before, and I'd like to revisit it and make sure we fully understand it. Then we'll use this picture to explain impulse transmission. The neuron itself is this black line in the middle. And it doesn't matter if we're considering a sensory neuron or a motor neuron, the picture is exactly the same for both. If this were a sensory neuron, this could very well be the dendrite, because that's the long part of the neuron that's in the mixed nerve. If it's a motor neuron, this would be an axon. These purple features are Schwann cells. The combination of Schwann cells make up what's known as a myelin sheath. And it's a covering of the long processes. Remember that these nerve parts are traveling in a mixed nerve perhaps, and there's many of them, and some are taking impulses in one direction, some are taking the impulses in a different direction. And this myelin sheath, made up of the Schwann cells, then protects the nerve cells from each other, it keeps them separated from each other, and it prevents them from what we call communicating with each other. Now this communication is what our topic is in this video. One other feature on here is these spaces in between. Those are the nodes of Ranvier. Now for this video, I want to just deal with what's going on at the membranes in the nodes of Ranvier, because the same changes, the same features do not take place underneath the Schwann cells, where this Schwann cells surrounding the neuron fiber. These changes do not take place there. So we're only dealing with what's going on at the nodes of Ranvier. So I'm going to take this little bit here and I'm going to draw it bigger. So this is perhaps a Schwann cell here, another one here, and this is the node of Ranvier in between. This section of a neuron is polarized. These neurons are polarized by the presence of ions. They're different concentration of ions. What we have on the outside is a lot of sodium ions. What we have on the inside is a lot of potassium ions. Plus, there's some negative ions, and we'll represent the negative ions by chlorine, because chlorine is a negative ion and it is present there. But you can see that the differential distribution of these ions makes the outside positive with respect to the inside. So what we have on the outside is a positive charge relative to the inside. Now this was first discovered, the fact that it's got a polarized membrane, when scientists were looking at the giant axons that are in the huge squids, the giant squids. And because they're dealing with axons, the vocabulary became axon vocabulary. And the same terms still apply for sensory neurons though, because if it was a sensory neuron, like I say, that'd be a dendrite, but we still use the terms like this. Membrane is called an axomembrane. Simply, it's the membrane of the axon. And the fluid inside here is the axoplasm. Again, that's cytoplasm of an axon. So there we have it. This is what's happening when nothing's going on in the neuron. this is what we call at rest. So we have a resting potential. Now, potential deals with voltage. And recognizing that there's a difference in ion charges here, there is a voltage difference. And when the scientists that were looking at the giant squid axons checked that out with an oscilloscope will give you a reading of the voltage difference, they found that at a resting potential, let's say this is the voltage here, and this is time, at a resting potential of about negative 65 millivolts. When this thing isn't doing anything, that means that the potential energy that's involved here is negative 65 millivolts at rest. That's the resting potential. And that's all fine and dandy. Now the next part of this story has to deal with the recognition that there are specialized membranes, there are specialized proteins in these membranes here. And this membrane, of course, it's round, it's circular. I'm just drawing it in cross section so that I can represent things. In order to represent them better, let me erase some of this writing, and I'll even erase the charges. We know that it's negative on the inside compared to the outside. I can leave axoplasm there and axon membrane there, that's okay. Now there are proteins present in here. And I'm going to represent three. I'm going to draw them slightly differently. Three different kinds of proteins play a very, very important role in this story. When nothing is going on, there's an abundance of sodium ions on the outside, an abundance of potassium ions on the inside, coupled with chlorine, which sort of means that this has a more neutral charge, and the positive nature out here means that the inside is negative compared to the outside. So that's what this is. This is the inside compared to the outside. When a stimulus is provided, whether or not the stimulus starts at a receptor on some end of a sensory neuron, or it's coming from another nerve cell, or even if this cell gets damaged or hammered in the middle, something disrupts this balance. And what happens is when a stimulus is strong enough, sodium will enter. And when sodium enters, what it does quite simply is it begins to reduce that negative nature, the negative nature inside compared to the outside. The positive ions are coming in, so this becomes increasingly positive in here, becomes more positive, and in respect, this outside becomes more negative. So in looking at it on a graph, what happens when sodium is entering is the negative nature of the interior of the axoplasm becomes less and less and less and less. It'll pass zero, and it actually, as this continues to flood in, it'll go up to about a positive 40. Meaning that the inside, the axoplasm, the cytoplasm inside the cell is actually positively charged compared to the outside, because there's so many sodium ions in here coupled with the potassium that are already there. That particular protein that I'm representing by a circle is, where's my blue book here? This one I'm representing by a circle is called a sodium gate. Proteins are sometimes called gates when they allow ions through. And if you recognize here, sodium or potassium, they're not free to move through membranes because the membrane interior is non-polar, and these things being charged, they would have to pass through a protein. So this is a protein, it's called a sodium gate. Okay, so that's the first thing that happens. We've actually changed the polarity here. We have polarized or depolarized. This is polarized to begin with. We've depolarized the membrane, because the polarity of the resting potential is negative 65 on the inside compared to the outside. Now, at this point here, the inside compared to the outside is positive 40 compared to the outside. So it's depolarized. And what happens to cause depolarization is sodium, what we say is that sodium floods in. Now, I have to move to another one of these, so I'm going to take this off for a second. I'm going to move it over. In fact, I'm going to move it all the way over to here, the sodium gate. This depolarization can only go so far because these other proteins are sensitive to electrical changes. This next protein that we're going to mention here is called a potassium gate. And the potassium gate is affected by this depolarization. In fact, what happens is the potassium gate changes shape and the potassium ions can move to the outside. So they do exactly what the sodium did, but in reverse. So what happens next, obviously, is if you're taking the potassium ions and you're flooding the outside area around the axon with potassium ions, that's going to reduce the positive nature, and it's going to sort of restore the polarity. And that's exactly what happens. This is the maximum depolarization, and this, where this potassium is coming in, or out rather, this is repolarization. So we've managed to get back, see, I've drawn that in here, we've managed to get back to resting potential. There's a bit of a blip here, I'll explain that in just a second. So our resting potential of minus 65. So the electrical nature of the axon has been restored because the sodium ions have gone out and the potassium ions have come back in. So everything's fine and dandy, with one exception. The ions are in the wrong place. We started out with an excess of sodium ions outside and an excess of potassium ions on the inside, but now what we have, because the sodium ions went in and the potassium ions went out, we have an abundance of sodium ions inside and an abundance of potassium ions on the outside. Except the resting potential has been restored. The ions have to be put back in the right place, because if you needed to pass an impulse along this cell another time, it wouldn't work, because the ions are already in the wrong place, and the first thing that's supposed to happen is that sodium is supposed to flood in, and it can't do that right now. So that's where the third protein comes into play. The third protein, I don't have enough room up here to write it all out, so the third protein is a sodium-potassium pump. I colored that one in. Sodium-potassium pump, and it literally pumps sodium out and potassium in. And I say pumps on purpose because the sodium-potassium pump requires ATP. It requires the expenditure of energy. So there has to be mitochondria associated with these cells. The energy has to come from someplace. So these ions are flipped back into their original position. So once we have the resting potential restored, we have the ions in the correct places, another impulse can be transmitted along this nerve cell. One little comment here, these sodium-potassium pumps don't pump these things equally. They pump 3 sodiums for 2 potassiums. So it's sort of a 3-to-2 ratio. And the balance seems to be awkward, but sodium ions do tend to leak in a little bit. And because of that, there's maybe more inside than there should be, so that 3 can be moved out, 2 get moved in like that. 2 comments. One is this wave of changed polarity that travels along this axon, this business in here, this is the action potential. Resting potential is down here. Action potential is the wave of changed polarity. And I say wave of changed polarity that travels along because when sodium comes in here and changes the polarity, that disturbs the proteins that are in the membrane adjacent to this. So it'll actually open sodium gates here, and it'll open sodium gates in the next part, and the next part, and the next part. So the fact that you get this happening in one spot along the axon disturbs the protein so that it happens in a subsequent place and in a subsequent place. So it moves itself along. And I can't say it fast enough because this stuff goes really, really fast. How long does it take an impulse to travel down your arm? This is lightning fast. So it propagates, or it moves along the axon simply because the disturbance in the ion changes, the electrical changes, disrupts the resting potential, disrupts the configuration of those gate proteins. The other thing, this was action potential, a term that's applied where sodium is moving in, which is the beginning of the impulse. This is an action front. And now I get a chance to describe more of the importance of the Schwann cells. The Schwann cells cover the whole thing. So what you get is the action potential occurring here. That disturbs proteins all the way along, but it'll disturb the proteins here. The ions can't move where the cell's covered by the Schwann cells. So the ions not moving here will move here and here and here, not underneath those cells. So what you get is an impulse that takes place there, there, and there, and it appears to go boom, boom, boom like that. And it's referred to jumping from node to node, is the way it's described, as the impulse jumps from node to node. It's not jumping. It's not leapfrogging over the cells or anything like that. It's just moving from node to node to node. And the term that describes that is saltatory transmission. This is saltatory transmission. And saltatory transmission is quite simply jumping transmission. That's what that is. That's what that describes. So there you have it. That's how an impulse travels along a neuron, or sometimes we say within a neuron. It jumps from node to node. It moves because of the displacement of ions, and there's a sodium-potassium pump using ATP to put the ions back into position to restore resting potential so another impulse can travel. What this doesn't do, though, is it doesn't tell us what happens when this gets to the end of the nerve cell. Because out here at the end of the nerve cell, there's either another nerve or there's a muscle or a gland, there's an effector of some sort, and we have to have some transmission story, some way to represent what happens when the transmission gets to the end of the cell. That's the topic of the next video. Thank you very much.